Welcome, everyone, to our IndieBio produced event, New Human Health Frontiers. My name is Julie Wolf, and I am the CSO here at IndieBio's New York program. I'm so pleased that you could join us today to meet three of our amazing Class 3 startup founders who are working in important areas of human health. Now, before I introduce our founders, this may be the first time that you've attended an IndieBio event, and so I want to take 2 minutes of your time to just explain what it is that we do here at IndieBio. So IndieBio, for those of you who are unfamiliar, is the world's leading biotech startup development program. We help startups to grow faster. Now, what do? We work with scientists who have a deep knowledge in a specific area of biotechnology and help turn them into entrepreneurs. How do we do that? Three main facets comprise the IndieBio program. One is the investment. Up to $525,000 is made available to help these startups move faster in order to get to market quickly. We have a 4-month mentorship-based program, during which time the startups will make different scientific, technical, and business milestones to help set them up for a successful next round of fundraising. And finally, we have a lab space, which is made available to our startup founders rent-free so that they can work on those technical milestones. We're always looking for the next frontier companies, and you can see the URL indiebio.co/apply if you're interested in joining us. Now, when we work with different biotechnologies, we work with people across the gamut using biology as a technology in both solving both human and planetary health problems. Today, of course, we're speaking with those that are working more on the human health side. But if you are interested in additional information about some of the other Class 3 startups, please join us at the end of the month for our demo day, which will be held also online at noon, same time in about a month. And we have additional sneak peek events, much like today's format, where you can learn about the startups in our Class 3 who are working on sustainable food systems and biotech infrastructure. And so with that, I'd like to focus on today's human health frontier startups. And I would like to first start by introducing today's panelists. I'd like to welcome to the stage Dr. Mehdi Javanmard, CEO of RizLab Health. Hi, Mehdi.
Hi, pleasure to be here.
I would also like to welcome Dr. David Bravo, CEO of Pannex Therapeutics. Hi, David.
Hi, thanks for having me here.
And thanks for joining us all the way from the southern tip of Chile.
Yay.
And I'd also like to welcome Dr. Harvey Tian, CEO of Inso Bio. Hi, Harvey.
Hey, glad to be here. Thanks.
All right, so for the audience, today's format is going to first feature an introduction to the different companies and what it is that their scientific products are. And then we'll jump into a panel discussion where I will ask a few questions of our panelists and we can learn more about their inspiration and their insight into these various areas of biotechnology. And then finally, at about a quarter till, in about 12:45, we're going to jump over to have some networking in the hop-in networking section. So without further ado, let me introduce Dr. Mehdi Javanmard to give his startup pitch for RizLab Health. All right, Mehdi.
Okay. Hi, everybody. I'm Mehdi Javanmard, co-founder and CEO of RizLab Health. And today I'm here to talk about the next pandemic, the pandemic of antimicrobial resistance, and how this pandemic is going to be orders of magnitude more devastating than this never-ending pandemic that we're living through right now. But more importantly, I want to talk about RizLab's front door solution to preventing this pandemic from happening. The reality of the matter is that antimicrobial resistance is growing, while antimicrobial drug development, on the other hand, is slowing. Now more than ever, antibiotic stewardship is of utmost necessity in order to prevent antimicrobial resistance while improving patient outcomes. The key driver of antimicrobial resistance is the overuse of antibiotics, and the key culprit driving overuse of antibiotics is the treatment with antibiotics of viral infections that have been mistakenly diagnosed as bacterial infections. And this is something that has been difficult traditionally in medicine. And so to prevent this next pandemic from happening, what's necessary is to provide physicians with the tools which they can make a rapid decision to confirm whether their patient's infections are truly bacterial or not. RizLab's front door solution to this problem is a microchip that we call the CytoTracker. The CytoTracker is the first ever lightweight, portable, affordable white blood cell analyzer that can within minutes quantify the total white count, the neutrophil count, and the lymphocyte count, and discriminate and determine whether the patient's infection is truly bacterial or not. The CytoTracker consists of a reusable, fully electronic reader, along with a disposable plug-and-play microfluidic test strip. The key heart and soul of the CytoTracker test strip is a microfluidic chip that consists of a very, very thin channel with a diameter thinner than a human hair that has inside it electronic sensors that can detect cells as they flow in a single file line, one by one, detecting them, quantifying them, and recognizing them. The CytoTracker is protected by 3 pending patents, but is also based on peer-reviewed technology that originated out of my doctoral thesis at Stanford University over 15 years ago, and was further developed over the last 7 years at Rutgers University. Now, the way that the CytoTracker discriminates between viral and bacterial infections is by looking at standard features that are well recognized and well established per standard of care, like the total white blood count, the absolute neutrophil count, the lymphocyte count, and also biophysical features like the electrical cell diameter. In addition to that, though, the CytoTracker extracts 8 proprietary features with which it feeds these all into an AI-powered classification engine, which looks at the unique signatures that each infection type has, and then makes a decision as to whether the patient's infection is bacterial or not. Now, I know the last time that we saw a movie involving a drop of blood and Stanford University, the movie didn't end so well. But this time is different. We have just completed a multi-site clinical study at Rutgers Robert Wood Johnson Medical School and Baylor College of Medicine involving 200 patients, where it was shown that the CytoTracker was 99% accurate in quantifying neutrophils and over 95% sensitive and specific in correctly detecting bacterial infections. Now, this is a competitive space, but the CytoTracker is unique. It's the only hematology analyzer that doesn't need to be used in a lab, and it also has the ability to discriminate between viral and bacterial infections. Now, because the CytoTracker is fully electronic, it can be manufactured very inexpensively, much similar to consumer electronic devices that we're all accustomed to. And that allows us to offer the CytoTracker at a price point that's significantly lower, at least an order of magnitude cheaper than existing hematology analyzers. Recurring revenue will be obtained from the sale of test strips, and providers can be reimbursed through existing CPT codes. We've tested the market, and within 3 short months, we've already received 9 LOIs showing that there is a true need for the CytoTracker in the clinic. Now, the hematology analysis market has a size of $6 billion globally. However, there's a much larger untapped market opportunity for antibiotic stewardship. I mean, essentially, think about it. Every physician that has this dilemma as to whether they need to prescribe antibiotics or not is a potential customer here. And when one considers that there's 2.5 billion prescriptions made per year and the number of tests that translates to, we're looking at a global opportunity of at least $28 billion. Now, this is a challenging problem, but we have the right team in place. Our technical team has over 30 years of experience in developing diagnostic technologies, That coupled with the fact that we just entered a partnership with Hacks to co-develop the CytoTracker, and we'll be leveraging their experience in product development of electronic hardware, all coupled with strong business expertise and clinical development as well. So our next step is to essentially manufacture devices and test strips so that we can perform a 510(k) study to obtain regulatory clearance so that we can launch our product by mid-2023. So join us in our journey to combat antimicrobial resistance and prevent the next pandemic from happening. Thank you.
Thank you, Mehdi. What a great overview of RizLab Health. Next, we'll hear from Dr. David Bravo, the CEO of Pannex Therapeutics, to tell us about one modality to treat chronic pain and potentially others as well. David, the floor is yours when you're ready.
All right, then. Well, I'm David Bravo, the CEO of Pannex Therapeutics, and we are a preclinical stage biopharmaceutical company accelerated by IndieBio in New York. And we were found to be focused on the developing of life-transforming therapies based on blocking Pannexin 1 channel. This new class of drugs is based on the discovery that I made on my PhD that Pannexin 1 channels plays an important role in the beginning and the maintenance of many diseases. Why does this happen, and why is Pannexin important? Because Pannexin releases ATP outside of the cell. Why is this important? Because ATP is supposed to be inside of the cell, right? But when there is a damage, or when there is an infection, this cell is breaking down. So this ATP that is inside of the cell now is outside of the cell. So this ATP is kind of a danger signal that says to everybody, there is a problem here. So what Pannexin does in pathological stages is to release ATP outside of the cell. And outside there is ATP receptors that receive this ATP outside of the cell and start many diseases such as chronic pain, as well epilepsy, opioid addiction. So this ATP comes out of the cell is the problem that causes migraine, causes neuroinflammation, stroke, autism spectrum disorders, multiple sclerosis, even Alzheimer's and other diseases. So we are here at Pannex Therapeutics. We are developing the first-in-class selective Pannexin 1 channel blocker to have this, to block this channel and have one pill for 9 different indications. And we are starting and focus on chronic pain and opioid addiction. Why? Because my mother-in-law, the abuela of my daughters, has chronic pain. And she started to take opioids and she felt better, but she started to become addictive and we almost lost her for addiction. So we changed the drugs and we use non-opioid drugs for chronic pain. And she now lives with a lot of side effects and with pain as well. So like her, there is 100 million of Americans suffering this condition, and this costs for the system more than $600 billion. But the problem is not that. The problem is that the best drug that we have, that is the opioids, kills 1 American every 15 minutes for opioid overdose. And the non-opioid drugs fails in almost 50% of the cases in reduces chronic pain. So we have here an urgent need for new therapeutic alternative for this high unmet medical need. So for this, we have developed the PX001 or the PNX3, the first-in-class selective oral Pannexin 1 channel blocker. What does it do? Well, as I mentioned, Pannexin 1 channel blocker, when there is pathologies, puts ATP outside of the cell, and this ATP causes chronic pain and opioid addiction. I discovered this on my PhD thesis and in 2014, and other researchers have discovered also that this ATP is widely important, and Pannexin is important in many diseases, such as those I mentioned. So what essentially the PX1 does is to block Pannexin 1 channel and inhibit this ATP release in order to solve those chronic pain and opioid addiction and prevent this opioid addiction and this chronic pain to happen. So this is a brand new mechanism of action. There is no other painkiller like this one. And this, we can call it the first anti-addictive painkiller. With this compound, we have our first results. And we have we found that the ATP release is reduced in 97% in brain cells, and also this molecule crosses from the blood to the brain, and it doesn't cause any harm to the brain cells. But the most importantly, our first hit of this PNX3 in vivo relieves musculoskeletal chronic pain in rats. And what we can see here, when they have a high score of pain, when there is chronic pain in animal models, but the PNX3 is able to reduce this pain at the same level of gabapentin. Gabapentin is the biggest player on market, and this drug by itself has a market of $3 billion in annual revenues. So, but the important is that we can make it with 10 times less dose, which means less than the competitor, less toxicity and less side effects. So based on that PNX3, we build up a library of new molecules that were checked for novelty in big databases in order to be sure that these are new chemical entities. They are novel molecules, and we also check it in computer for the see if they can be used as a product, and also predict this toxicology. And then with those molecules, we use the actual human Pannexin 1 channel atomic resolution in the computer, and found if our molecules can be docked or can be coupled to the specific part of Pannexin to block it. And then we have 23 lead compounds ready to go to be synthesized, and they have been synthesized now. We have 11 molecules synthesized, and they are currently being tested in different countries, in different animal models, in vitro and in vivo. So this is where we are. We got here thanks to support of many the previous investors, and of course thanks for the support of IndieBio. And with that money, we've been able to synthesize those molecules, work on our patents, and make first proof of concept in animal models, in order to, in the future, having our lead selection and go from those 23 molecules to just the first molecule to go to the FDA and the approval for having testing in humans in early 2024. Well, as you know, why this is interesting, because the chronic pain market is huge, it's underserved, and is growing with and the non-addictive, the non-opioid drugs are growing with a CAGR of 18.3%. So there is a lot of space to grow here and to penetrate this market. And we know that even in clinical studies and in the market, there is a lot of competitors where we have the potential to be the first anti-addictive painkiller in order to prevent this opioid addiction crisis and treating chronic pain at the same time. But also we have a new mode of action, different from all the other compounds on the market. And since Pannexin is mostly doesn't have many functionalities in normal people, but it has, when there is problems, when there is pathologies, we expect very low or no side effects from our compounds. And we already prove our effectiveness, so we can say that we can be key players in the future market. And for that, I count on a very experienced team. I've been dedicating myself as a co-founder 10 years of my life studying Pannexin channel, but now my other co-founder, Dr. Thomas Gerlach, my chief of strategy officer, he has dedicated 30 years of his life in developing, in working on global pharma development and strategy. He got a CRO, a startup like mine called Octillion, from the preclinical studies to the market and sell that company in $33 billion. So he knows the entire process. And Dr. Gerhard Gross, my chief of scientific officer, has 14 FDA-approved molecules designed by him, since he was the head of preclinical studies in many global pharmaceutical companies. So with this strong team and a vast panel of experiences, academics from different areas, we are working on this ambitious endeavor to develop the first anti-addictive painkiller to save your relatives' lives, and develop the first pill for 9 different indications. Thank you.
Great overview, David. Thank you. And what an important first problem to be attacking. All right, our final introduction to a company will be by Dr. Harvey Tian, CEO of Inso Biosciences. Harvey, we're very much looking forward to hearing about your new technology.
Thanks. Yeah, great. Hi, thank you all for joining us. My name is Harvey. I'm the CEO of Inso Bio. So we are a Cornell University spin-out, early stage biotech company, and we're developing hardware solutions to become the next de facto way of preparing genomic samples. An example of something that our technology has been able to do, as you can see in this picture, is take a single finger prick droplet of blood, process it on our technology, which has these microfluidic disposable chip cartridges, process that within about half hour, and then send it off and sequence the entire human genome from just that single droplet of blood. So that's just a demonstration of the capability. So what we are really trying to address is the lack of scalability when it comes to preparing genomic samples. And genomics as a field has been improving exponentially over the last 10, 15 years, with sequencing costs dropping faster than Moore's law, the number of data analysis pipelines sort of exponentially exploding over the last decade. But the way that people are handling biological samples hasn't really been innovated upon in a few decades now. And so that's what we'd like to attack. An example to give a little bit more context to this problem, I'll run through a use case with Sarah. So Sarah is a genomic core facility director at a top US institution. She could be an example customer, but she could also be a genomics core director at a clinical lab. She could be a genomics core director at a R&D facility at a company. Essentially, people like Sarah have about half a dozen highly trained techs that work for her, and they prepare samples regularly for genomic sequencing. But even then, they struggle to prepare high quality samples in a reasonable amount of time. And this is especially true for the newest form of sequencing called long read sequencing, which uses longer pieces of DNA to create new insights into areas like de novo genome assembly, cancer genomics, and agricultural genomics. So how we're going to be able to help solve Sarah's problems is we're going to take all the complexity and friction that's involved with preparing genomic samples, handling biological material, and streamline that into a single automatable system that can deliver higher quality DNA in a shorter amount of time. So this is possible through technology that we invented at Cornell University through my PhD. This technology has since been exclusively licensed from Cornell, and what it does is it utilizes tiny microfeatures that we call a micropillar array. And this micropillar array can perform two very key unique functions. First, it can extract the DNA purely physically based on size, almost kind of like how the prongs of a fork can pick up spaghetti. So there's no in-channel functionalization, there's no binding events, it's purely a physical capture of DNA. And then secondly, the reason that we're called Inso Bio is because we keep the DNA in solution throughout the entire processing, in solution. That's unique, as no other technology is capable of doing that. They generally have to dehydrate and rehydrate the DNA through multiple precipitation steps, which then causes the DNA to be damaged and reduces the sequencing quality. So that's what allows us to have that 30% better sequencing quality. We also combine the extraction and purification step into a single simultaneous step, which has allowed us to complete the workflow in about 10 times faster speed with about 10 times less hands-on. So the technology was developed at Cornell, again, under the Craighead Research Group, and Professor Craighead is one of our co-founders, but he's also a co-founder of PacBio, which is one of the world's now leading sequencing companies behind Illumina. I completed my PhD with Professor Craighead, and I've spent about 10, 11 years now working on the underlying technology. And our third co-founder, Adam, was originally our academic collaborator, and our entire co-founding team had worked together closely on this for about 3 years before starting the company. Now, when you look at what other sort of technologies are out there and are available to users like Sarah, it's generally that they have to use at least 3 categories of technologies to complete a single workflow. Whereas for us, we can combine all of the steps from those different categories of technologies and into a single system. And we can reduce the amount of sample loss and improve the quality of the sample by doing so. And not only that, we remain competitive against the best in class of each of the categories of technologies out there. So we've been fortunate enough to work closely with PacBio early on. We've received encouraging data in collaboration with them. We've received the support letters from industry partners, potential customers such as Corteva, which is a large agriscience company. And we've received letters of intent to purchase our system, outlining over $500,000 of potential revenue in the first year of market entry. So with that, I'd like to close by outlining sort of the greater vision of what we're trying to build. Essentially, we have a platform that's a hardware that can be applied to many areas of biological sample handling and sample preparation. And what we'd like to go after first is the area of long-read DNA sequencing sample preparation. But this is not the only area that our technology can be applied to, and not the only area that we have data for. Our current existing NIH grant is for using our technology, the same technology, for infectious disease processing. And then we had a previous NSF grant, which uses the same technology again for single-cell multi-omic processing. Ultimately, what we'd like to address again is the lack of scalability when it comes to handling biological samples. And when we can improve that, then we can catch up to how far genomics analysis and genomic sequencing has come already. And by doing so, we hope to enable those applications of tomorrow. So yeah, thank you.
Thank you so much, Harvey. Thank you for the great overview of your Inso Biosciences, and to Mehdi and David as well. So I want to start the Q&A portion, and I think the first thing that I'd like to ask is the key insight that each of you had that made you think, I think I've got something here. I want to pursue this and create a company based on this technology. And I'm going to turn first directly back to Harvey, because I know your origin story, and I think it's one of the greater serendipity events that have happened in this cohort of IndieBio founders. So Harvey, could you please tell us what your insight was that you had a novel technology, and how that inspired you through your work to create InsoBio?
Yeah, absolutely. Thank you for asking that, Julie. It is a funny story, and it was actually our first year, well, my first year here as a graduate student at Cornell, and I was working with Professor Craighead and in his group, along with a few other postdocs, and we were working on this underlying technology, and we were originally using those same what we call the micropillar array structure. We were using that simply to separate out where the cells were versus where we loaded magnetic beads, which we were intending to use to capture all of the DNA. And then when we lysed all the cells in there and tried to capture the DNA onto the magnetic beads, we kept finding over and over again that we had nothing. And it really confused us for quite some time. For about a month and a half, we were really scratching our heads, trying to figure out why loading more and more cells was still not producing any DNA caught on those beads. So finally, we decided we'll just have to look inside the channel and figure it out. We put in dye, we stained things, and then, lo and behold, all of the DNA was caught on the actual micropillars themselves and not on the beads. And so, at first we were like, oh crap, that's not good. But I think from there we sort of thought about, is there a way that we can utilize this sort of, as a feature, not as a bug? And that's kind of how the entire idea around using this as a highly efficient and highly capable way of capturing and processing DNA originated.
Excellent. Yes, it's a great story and a nice origin tale for Inso. Mehdi, I'm going to turn to you next, because I know you've been working on miniaturization for your entire career, but you haven't always been focused on the problem of differentiating, cell responses or cell types. So can you talk about what your insight was that led to the formation of RizLab?
Yeah, great question, Julie. So essentially, as I mentioned in my introduction, when I was a doctoral student at Stanford, we worked on miniaturization, building small, tiny labs that can detect microbes, that can detect different types of mammalian cells, but doing it all electronically. And then over the last 7 years at Rutgers, we continued to perfect and fine tune and optimize the technology and our ability to detect these different cell types. And then the challenge was, though, the footprint of the instrument was still large. But in 2017, essentially, what we figured out was not only how to make small chips, but to make everything small, the electronics and everything, interface it, make a system that had no moving parts. And what happened simultaneously, or at the same time as that, in parallel, was that we found that there's a gap in the market. Essentially, we found out that there's no portable CBC, or complete blood count analyzer. And talking to various stakeholders in the healthcare ecosystem, we found that there's a real gap, and there's a really good product market fit. And so that was essentially what led to the spinoff of RizLab Health out of Rutgers University.
Oh, very interesting. And David, you mentioned discovering the PNX3 and you were studying pain in an academic setting. Now, a lot of basic researchers will discover this mechanism, but not necessarily follow up by looking for compounds that will interact with that target. When was it that you thought, okay, I think I'm going to pursue this as a therapeutic modality?
Well, in the very beginning, when I started my PhD, I knew that I had to find some solution. So the answer to that is when I decided to make my PhD thesis on chronic pain, I was always thinking, okay, I will find something new and I'll pursue a solution on that regard. That was 2012, almost 10 years ago. And based on that, I started to make a deep understanding on this Pannexin 1 channel. But it wasn't until 2017 that I decided to say, okay, I will dedicate my time, my life, my focus and work on this performing pilot experiment and starting really from zero. I'm not a chemist, but intuitively I started to design molecules by learning and studying and getting advices, precisely because I saw many of my colleagues in Chile and in other countries developing groundbreaking solutions for chronic pain or other diseases that just are full of dust today on a desk or on a library. So I said that's unfair for the people that really needs this, like my mother-in-law, like my former patients they motivated me also to find a solution. So 2017 was the year of not only having this intentional idea, but do something and start this long work.
And I next want to ask about the market strategy. Like you, David, mentioned that you're going to go first for chronic pain, but that there are a number of different promising indications that would also be treated by this same mechanism of action, or maybe a similar compound. Can you talk about where you might see, of course, getting one therapeutic modality into the market is going to be a long road, but where do you see perhaps your next step, or what the largest, let's even just talk about what some of the larger patient populations are that would benefit from this type of medication?
Well, this Pannexin 1 channel is in mostly every chronic disease that has been described. So chronic pain, it's one of the biggest market, specifically musculoskeletal chronic pain. We're talking about $32 billion market to starting as a first indication. But nobody's able to count how big is the market on opioid addiction. And with our mechanism of action, we can prevent those people to become addicted to opiates, and that could be a very large market as well. But our most ambitious endeavor in terms of market size is by far cancer. We have been working on that side of research, and there has been also many research independently and peer-reviewed showing that Pannexin is highly important in different aspects of cancer, such as in the multiplication of the cells, but most importantly in the metastasis. So this is $100 billion market that we are pursuing to go in the near future. We are getting our first molecules to this way that we do know, but we are making alliances and growing team, going to oncology studies to go for that market in the coming months, and in the preclinical development and research, and hopefully in the coming years to support those patients as well.
Yeah, fascinating. Not only the chronic pain, I imagine the pain medication may also help those cancer patients, but also the disease itself. Fascinating. Wonderful. And Mehdi, you had also mentioned, multiple uses. You mentioned a lack of a CBC on the market, but that's not exactly what you're using the cytotracker for initially. Can you talk about how you'll find other applications for this portable, affordable white blood cell analyzer?
Yeah, great question, Julie. So essentially, as you correctly pointed out, point of care instant white blood cell analysis has numerous use cases. I talked about bacterial versus viral discrimination, cancer patients who have to get their chemotherapy doses regularly need to get their white blood counts measured. Schizophrenic patients, for example, on drugs like clozapine, need to get their CBC performed, their white blood count performed up to 3 times a week. There's numerous indications where this can have very high impact. Our focus, as I mentioned, is going to be initially on discriminating between viral and bacterial infections. Our go-to market is going to be clinical labs, emergency rooms, the ICU, also urgent care clinics and primary care clinics that have in-house accredited labs.
Oh, very interesting. And Harvey, I want to turn same question to you about InsoBio, because you mentioned having a number of LOIs, and some of those were in a human health panel, and yet you mentioned having a large agro company that is one of your LOIs. So can you talk about how really this is an application, of course, to things like genomics for human health, but really anything that has DNA?
Yeah, absolutely. So I think with the newest form of sequencers, which is the 3rd generation now of sequencers called long-read sequencers, they're really being applied in a lot of different areas. It's starting to see applications in the clinical realm, but it's not the only area that it's shown a lot of value coming out of the gate. The other areas is really in agricultural genomics. Lots of the plant breeding where larger genomes and potentially more mutations, these things are things that the long read sequencing actually has a huge advantage of over short read sequencing. And that's why we want to start there, but it's really not the only place that we want to go with this technology. We're sort of see ourselves as the superior way to really extract and handle DNA prior to all of the analysis, whether that is sequencing or that's some other form of analysis like optical mapping. So yeah, I think we'd like to see ourselves as a general ubiquitous upstream tool in front of genomic analysis, whether that's for human health, and that's eventually where we want to go as well, or for other areas like agricultural genomics, de novo assembly, science in general.
Oh, fascinating. All right, so I want to ask kind of a question that a lot of entrepreneurs are not asked during the pitch, a little unconventional, but what is we know at IndieBio that building a startup is hard work. It takes a long hours. It takes constant, looking deep into your soul and making sure the motivation is there. And I want to know what your motivation is, what you found most rewarding along the journey. And Mehdi, I'm going to turn it first to you because I think I've started with you yet, to ask what is it that keeps you motivated in working so hard to continue to build RizLab Health?
So this is an excellent question. I mean, I've spent my whole career working on making miniaturized tiny labs, and what really excites me particularly about RizLab and motivates me every day to drive the company forward is this feeling and this knowledge that one day our device will be used in clinics. It's going to be used to improve the standard of care for patients and relieve the suffering that they can go through, decrease mortality. All of these things are definitely what keeps us excited and motivated every day.
Oh, wonderful. And David, I know you mentioned the abuela of your grandchildren, or of your children, and others that you've worked with as a clinician. Is that what motivates you, or what is the thing that you look to that keeps you going in the day to day?
I mean, it sounds very idealistic, but it's actually have a better world to live with better quality of life. For me, that it sounds very ambitious, but I wake up like this with the sensation every morning, like I can do something meaningful to help people. I grew up seeing my parents picking up people, addicts, from the street to help them, anonymous people, to help them to get recovered and go to those centers of rehab of addiction. And I was a very young kid. It was like shocking for me to see how committed they were to serve the other and to change their life and change and improve their quality of life. So that wakes me up in the morning. And but by far my biggest motivation is to make a better place and a better world for my daughters, which are my biggest driver and my biggest motor to do everything in my life, starting from waking up to work and do everything, having a solution for them, for their friends, for the world they will live in the future, and of course make them proud of her dad as well.
Yes, and I can certainly attest to having worked with you through these past 4, 5, 6 months that you are definitely trying to do good in the world every day. Harvey, same question, motivation. What is it that keeps you, at the grindstone every day, long hours, lots of meetings, facing challenges, et cetera?
Yeah, I think it's also a great question. I think for me, at least personally, I think it's largely understanding or sort of hope for what the future should be or could be. I think certainly the way that I see it is the world that we're building towards and living in, and this is not my quote, but I spoke recently with someone named Andrew Hessel in charge of the Genome Right Project, and this guy was very amazing. He put it very eloquently that the world around us, traditionally we've seen it as like birds and trees and animals and et cetera, and humans, but that's one way to see it. And another way that we're starting to see that world now is that it's all just code, right? We all have genetic code, it's genetic material. And instead of only seeing it as those things, we can also see it as code that's not only starting to be able to be read, but in some cases we can even interact with and reprogram that code. So I think it just makes for an extremely exciting future. And I just don't think that that is possible without some improvement or some dramatic improvement and increase to the efficiency of how we interact with that biology.
Excellent. Yes, well stated. Thank you. Andrew Hessel is a very smart man. So final question for the panel. I'm going to go around and ask you to very briefly describe how you think your field is going to be changed in the next 5 to 10 years. And as you're thinking of your answer, I'm going to turn first to Harvey, because I know you've spoken about genomics and the future of genomics, which is a future that is yet to be written in the code of DNA. But I wonder if you could talk briefly, Harvey, about how you think the future of this very broad field will begin to unveil itself.
Yeah, I think in my mind, going back to the programming and the analogies to the sort of computer revolution, I sort of, in my view at least, see this as a very nascent but similar journey to where the computers took our society and our species. I think we're very much in the early days right now, where DNA sequencers are extremely large, they're extremely expensive, sort of like old mainframe computers used to be. They're limited in certain ways. The ways that we process and interact with biology requires a highly trained scientist, just like you used to have to be a computer scientist in order to interact with those mainframe computers. And what the world, I think, eventually will move towards, and what our field especially definitely will move towards, is integration, vertical integration. So I think right now, some scientist out there is performing biological sample handling, then they put it onto a large sequencer, sequences it, and then some other data analysis expert is looking at that raw data and converting it into meaningful information for the ultimate project, whatever that is. I think in the future that will all happen streamlined very quickly on a single system, which we're still building that towards that right now, I think, as a society, And I think a very important part of that is, how do we create this box, so to speak, that can process everything together. And a big component of that is the front end, handling the biological sample in the first place.
Oh, what a fascinating worldview. I could ask many different follow-up questions, but I'll leave that for our audience to ask you at the networking section, which we'll jump into in a minute. All right, I'm going to jump to Mehdi and ask how you see RizLab and the technology that you're building with the CytoTracker and perhaps even other tiny labs being something that will change the world from the patient experience, from the clinician experience, and just the greater society.
Yeah, that's a great question, Julie. So I actually want to rewind back to about 3 years ago. You know, as an entrepreneur, you have to sometimes deal with the naysayers as well. I remember there was a physician that I was talking to, and this was before the pandemic, who said the only useful point of care test is glucose testing. And I think our centralized lab system is just fine as is. And then the pandemic happened. And, you know, guess what? Less than 2 years later, now, you know, there are you can do home-based COVID testing, right? Things that were like, you know, impossible to imagine, before the pandemic. And I think this shows, I guess it's one of the silver linings of the pandemic. And it shows that our mentality and our view towards diagnostics is changing. And I think our philosophy and vision at RizLab fits very nicely within that. I mean, what really excites me about technology is wearables, right? Everything, things are becoming wearable, portable. This idea of getting more data about physiological parameters, now also biomolecular parameters, what we're exposed to in the environment, how all of this is going to be fused together, I think is going to allow for an unprecedented, improvement in the standard of patient care, and can totally change how the healthcare system works. That really excites me.
Yeah, what a fascinating picture of the future, Mehdi. All right, David, last words go to you in terms of how you think the world will look different in 5 to 10 years, given the success of Pannex Therapeutics and the ability to successfully treat patient pain.
Well, it will change in the terms of knowing that today it's impossible to think about one pill for different indications, and this is what our biggest bet is, to have one mechanism of action on one set of pills to treat many diseases, because as we know, these are chronic diseases, and the chronic diseases are increasing in our society, and the people live with more than one chronic disease. Just the example that you already gave, people has cancer pain and also cancer. So what if I can treat, instead of giving 10 pills, give just one to treat both? Is that possible? And we believe in the coming years it will be possible. But not only because of our strategy, but I'm not the savior of the people that is sick. Artificial intelligence and computational biology will be the approach to have even more accurate and specific multi-targeting new drugs coming in the future. And these are also goals that we are starting to look and pursue as a company, to use those strategies to have even less toxic, more specific, more powerful, with smaller doses, new compounds to solve more than one problem at the same time. This is the way we see the future, and we expect the future to become when we are on the market.
Wonderful. What a wonderful vision of the future. Well, I'd like to first thank all of the panelists, all of our startup CEOs. We know your time is very valuable, and we appreciate that you've shared your vision of the future, answered these questions, and given us a wonderful overview of your startup during this hour. Now, for those of you in the audience who have further questions for our panelists, we are going to close this main stage of Hopin and jump into the networking session, where we can have short one-on-one informational meetings. You can ask a follow-up question, introduce yourself, and hopefully enjoy meeting one another, as well as our panelists here today. All right, so thanks everyone. Thank you, Mehdi, for joining us today. Thank you, Harvey.
Thank you.
Thanks.
And thank you very much, David.
Thanks a lot. It's a pleasure to be here.
All right, we'll see you over in the networking section.